Simultaneous separations of lipid nanoparticle components using nearly monodispersed mixed-mode stationary phases. The recent FDA-approved COVID-19 vaccines use lipid nanoparticles, LNPs, to protect and transport mRNA to cells. Despite LNP drug delivery systems becoming increasingly common, analysis of LNPs is difficult due to the opposing physiochemical properties of their components. mRNA LNPs are typically analyzed over multiple methods to separate by different physiochemical properties. LNP component separation is important for quantitation and optimization of lipid composition for desired physical properties, such as surface charge and particle size. This experiment uses two stationary phases, a C12 and C18 arrow column, to separate empty LNP components within a single method. These MACMOD elosphere columns were selected for their nearly monodispersed particle packings, which enhance peak shape and efficiency by minimizing eddy diffusion. For the methods, the LNPs in the experiment were composed of SM102, an ionizable lipid, DSPC, a phospholipid, cholesterol, and DMG-PEG, a PEG lipid, in a 50-10 38.5 to 1.5 molar ratio. The solution was diluted to a tenth in a phosphate buffer. The methods for both stationary phases are the same, except for the ion pairing agent used for the C12 column. The MAX18 arrow column used formic acid to help with the resolution. MAX means that the stainless steel column was coated in a bioinert material that reduced secondary interactions, which can reduce sample recovery and cause peak tailing. Both stationary phases were able to separate the LNP components. The baseline increased on the C18 arrow column through the run. We suspect the rise in baseline is due to species contamination on the MS detector. As the IPA percent increases, it kicks off contaminants and the signal is shown for them. This is not visible in the previous C12 study because TFA is known to suppress ionization. Since then, the detector has been cleaned and the increase in baseline decreased. For the C12 separation, the ion pairing agent trifluoroacetic acid improved resolution by increasing the retention time of the charged species. Ion pairing agents interact with ionic compounds in the mobile phase, forming neutral ion pairs. This increases the hydrophobicity of the analytes, enhancing their affinity for the hydrophobic stationary phases in reverse phase chromatography. Initially, SM102 and cholesterol partially co-eluted. Reducing the TFA improved separation. Thus, TFA percentage of the mobile phase was systematically optimized from 0.1% to 0.01%. The diluted PEG lipid did not present a peak in the chromatogram. The low concentration and high polydisparity of DMG-PEG led to weak signal response, but was still detectable, eluting before the other components. Figure 4 shows the PEG lipid mass spectra of the C18 column chromatogram for proof of detection. For the MAX C18 arrow separation, the C18 arrow column provided a comparable lipid separation without the need for a TFA ion pairing agent. This column uses aromatic groups to provide ring stacking interactions with cholesterol, thus causing it to retain longer than SM102. Ion pairing agents are not ideal for liquid chromatography because they can cause column and MS contamination. They are also environmentally toxic. Thus, the C18 arrow method will be used for lipid quantitation and subsequently optimized for RNA and lipid separations.